And I'm happy to welcome William Bains as our first speaker, who is going to talk about searching for life in stranger seas, solvents other than water. William is associated with Cardiff University and MIT, among various other places. Thank you, William.
You. Okay, can you hear me? Awesome. Next. So I was asked to talk about alternative solvents for life. I'm going to start out slightly broader and say, what can we say about non-terrestrial life? There's a lot of speculation around that, about what life could be. It tends to start from the assumption that life will be different from life on Earth, rather than saying, and how different, it depends on your point of view. Rather than saying, what could it be from first principles? I think life does something really interesting. What life does is it takes input and turns it into more life. And what it turns it into does not depend on the input. So if you took a young Baines and fed him chips for 70 years, would you turn into an old Baines with severe cardiovascular disease, or would you turn into a potato? Now, yeah, you laugh because the answer is obvious. But this says something really quite profound about what life is, what we understand life is. And that is that life can select from inputs. So it can take inputs, take selections of compounds from it, convert those compounds into more me, and throw away the rest. And this means all life eats, poops, and grows. But also it implies there's some internal code that says what this life should be like. Not a pattern, but a code. And it also says that there must be catalysis to take the right chemicals in there with very specific transformations. Sorry, the chips are really dry. I bought them last night. Specific chemicals, and produce them very specific outputs. So catalysis is important, not for speed, but for specificity of reaction. And that implies all the chemistry, including making the catalyst. And so in order to avoid an infinite regress, you must end up with polymeric catalyst. And that means you must end up with a dense, mobile solvent. Because if you try to dissolve polymers in air, they just fall out to the ground. If you try to dissolve them in solid, you form a solid solution, but the molecules that they catalyze can't move between them. So we come to a general rule about life, that life must have a dense, mobile solvent. And we come to this not by saying Earth has that, but by what we think life is. This is an example of a number of rules we're trying to develop as an overarching thesis with the work at MIT and elsewhere. We have a couple of others I might come on to. Now, on Earth, of course, the solvent is water. All life on Earth depends. And water's an amazing solvent. It's flexible. It's perfectly chemically done. It's probably the most common molecule in the universe after dihydrogen. It's present as liquid in all sorts of bodies. So there's a school of thought that says water is the solid function. And that's based on the solids cannot possibly exist. We still want to challenge that, say from first principles, can you say without other solvents would work in life? And in order to do that, you need criteria for looking at other solvents that isn't easy water. As we can, 4 criteria. The first is that says a calyx up there, sorry. So what's actually occur? Look, in a sense, it's not fantasy material, but in a sense, it's occur as a liquid form, really stably on a planetary or near a planetary surface. Why? It's simple ecology. If you have a life form living on a planetary surface, say it makes its own salt. Fantastic. It's making some weird salt. And then it gets injured, or it runs out of food and loses that salt. What happens is that it permanently dead. And that persistently is permanently dead. Unless if we re-wet it period, like salt from my bar. So the salt has to be in it. That, of course, is temperature pressure independent, so whether salt effects of the environment is what's an accident. They must be stable enough to flutter chemistry reaction with rocks and salt, and we're talking about this as people. So currents. Solvation, there's all things, there's all things too. But not everything, because if the water was perfect solvent, everything, you would be puddled, so you would be freeze. So you need things that dissolve and soluble and not solvent. And also, ideally, things, molecules, potential molecules that are both soluble, bonding, and insoluble.
which would race up similar things like cell membranes, DNA, double helixes, proteins, and so on. And related to that, stability. The molecules must be stable to have all these functions. What sufficiently stable is depends on what functional molecule is. So for metabolites, have glucose amino acid, probably hours to weeks. For DNA, centuries. But it must be reasonably stable. And a diversity of molecules must be stable. Again, deduce this from first principles without saying what that diversity is built on.
You need molecules in different shapes, so they interact in different ways, with different electrostatic fields around them, so they attract or repel each other in different ways, different bonding orbital structures, so they have different chemistries and reactions. And the reason for this is very simple. Doing this complicated biochemistry is very complicated. You need lots of different sorts of chemistry to happen. And there's a need for some self-consistency with that, which I won't get into because we don't have time today. But basically, doing this, turning small William into large, rather ill William, is a very, very complicated chemical process. You need chemical diversity to do that. So you need to dissolve things, they need to be stable, they need to have a lot of different things. And the last one is slightly more sort of terra-centric. We observe water is not just a solvent for life, it's an active participant in biochemistry in many, many ways. And this is one of the reasons people think the water is the solvent for life. Is this necessary? Can a solvent for life just be a solvent and all the chemistry happens between the molecules dissolved in it? We don't know. But it does seem to be plausible to suggest that having a solvent that could actively participate in chemistry would be a good idea. So 4 criteria: occurrence, solvency, stability, and chemical functionality. How do you actually apply these? Well, I've got 3 examples I'm going to whiz through very quickly just to illustrate. Thank you. Also a nice chap. Just to illustrate on there, how you do this. So liquid carbon dioxide. Now, on Earth's surface, carbon dioxide doesn't form a liquid. It goes straight from solid form, dry ice, into gas. But under sufficient pressure, a liquid phase of carbon dioxide will form. So this is the phase diagram of carbon dioxide. I'm going to give up on this. This is the phase diagram of carbon dioxide. Temperature on the bottom, pressure up the axis. And on the zone to the upper left for you, carbon dioxide will form a liquid. Below 32 degrees centigrade, which is its critical temperature, above which you form a supercritical fluid. And at moderate to some high pressures. And this is an example of this. This is a terrestrial example of this. This is the Champagne vent in Mariana Trench. And I don't know if you can see, but there are little blobs coming up from the surface there. Those aren't bubbles. Those are droplets of liquid carbon dioxide. CO2 coming up from volcanoes under this volcanic vent gets cooled by the ocean under the pressure low down in the ocean. It condenses into liquid. And then that breaks through the silt on the ocean floor because it's less dense than water and floats up the surface. So cold and pressure. Where would you get that? Well, you're really talking about something where you need lots of CO2. You want it cold. You want it like Venus, but cold. Could you do that? Well, there's a really interesting scenario where you can generate a planet of that sort. If you think of a red dwarf star, its luminosity is not constant with time. Our sun's luminosity has been reasonably constant since it was formed, plus or minus 50%, which is enough to freeze the Earth, but we won't worry about that. A red dwarf, a cool red dwarf like TRAPPIST-1, starts out very much brighter than its main sequence luminosity. And so if you have 2 planets orbiting around a cool red dwarf like this, in the early part of its evolution, the outer planet is nice and clement. You can have liquid water on its surface. The inner one is baked dry. It's like Venus or Mercury. By the time it's reached main sequence, outer planet is frozen solid. The inner one now is a clement temperature, but it has no water. It has no hydrogen species at all. They've all been evaporated up into space. So if that was a volcanically active planet like Venus, you'd end up with an atmosphere of a heavy atmosphere of heavy gases like SO2 and CO2, and you could have liquid CO2 oceans on the surface. So currents, certainly not on Earth, but in other environments, possibly. Solvation, CO2 is a moderate solvent, not brilliant. Stability, CO2 is quite unreactive. And so, yes, you can dissolve lots of things in CO2, and you cannot stably dissolve in water. And chemical functionality, unfortunately, is terrible because it's not very reactive and it's just dissolvable. So, liquid CO2. This is some illustration of how we go through these sorts of lithology. Liquid sulfur. This is really, so I tried to do some experiments with liquid sulfur in the lab. My PI told me to stop. Liquid sulfur is quite rare on Earth, but it does occur in volcanoes. It probably occurs in volcanoes and Io. The problem with liquid sulfur is the temperature range over which it is a suitable liquid for life. So the graph on the right is the viscosity of liquids. The grey line is sulfuric acid, which we'll come on to in a moment. The blue line is liquid sulfur. And just to give you some, this is in whatever is Newton seconds per meter squared, which most people don't understand. I certainly don't. So for comparison, this is the sort of viscosity of materials. And up to about 160, 165 degrees centigrade, sulfur is a nice runny liquid. So the bars on the right are the range of viscosities that we know that terrestrial biochemistry can function at. It might get a function outside those, but those ones we definitely know it can work at. Sulfur shoots up, and you've got basically trying to ask for life in toothpaste at sort of 180, 190 degrees centigrade. So it's too viscous for that sort of dynamic chemistry to be plausible. So in terms of occurrence, yes, you could have liquid sulfur, but it is in quite a narrow temperature range. It's not clear how you get that in a sort of planetarily plausible context. Solvation, pretty much unknown. Who dissolves stuff in liquid sulfur? I tried, it went brown, which addresses the lower one, stability. Sulfur is quite reactive. But as an active participant in biochemistry, sulfur is fantastic, much better than water. All the complicated redox reactions that life has to engineer with very complex metabolism, sulfur just does for you. So, pluses and minuses. Sulfuric acid. This isn't water with sulfuric acid dissolved in it. So battery acid is basically water with a lot of sulfuric acid dissolved in it. We're talking pure liquid sulfuric acid here. How would that occur? Well, the same cool red dwarf planet scenario as worked for CO2 could also work for sulfuric acid, except you don't need the high pressure. Volcanic outgassing of SO2 will result in sulfur dioxide and sulfur trioxide in the atmosphere, and if there's very little water around, then that will form sulfuric acid. And this is, of course, what's happened in the clouds of Venus. Is why they're made of sulfuric acid. Won't happen on Earth because, as the graph on the left shows, this is abundance of gases coming out of volcanoes on Earth, and by far the most abundant one is water. So not sulfuric acid on Earth. So occurrence, a reasonable scenario for occurrence. Stability, solvation, chemical functionality. I'm going to skip all that because Janusz is going to talk about it next. So that saved me 5 minutes, doesn't it? So this is the table we ended up drawing up, and it was real disappointment for me because water's at the top, and that's the only one with the green tick across all the columns. And what a shame, eh? Sorry, Ray, we can all go home now. Sulfuric acid down there on the third row down looks promising. Stability issues are still being addressed. Liquid carbon dioxide look promising. Across and chemical functionality, but we don't know if that's really important. A couple of ones people talk about a lot. Liquid ammonia, forget it. Liquid methane, forget it. Sorry, I'd love there to be life swimming around in the oceans in Titan, but it seems very unlikely. So what? I think the so what's quite important. There's, and I think it means we should not write off so-called uninhabitable planets as uninhabitable full stop. They might be too hot or too cold for water-based life, too little atmosphere, too dry for water-based life, otherwise there's no water. But there could be other solvents on those environments which fulfill the criteria for a solvent for life. So we might be looking for a large, warm Mars or a cold Venus, harboring very different sorts of life. I think there's another sort of so what though, which, yes, I do have time to touch on very briefly. So that's all astrobiology, and as two of the speakers said in the introduction, the public are really interested in this. They want to know, and we want to know, and to direct missions for searching for life and get on Netflix for documentaries. But there's a practical thing. If you can understand the rules around which the chemistry of life operates, you can start to understand what happens when it breaks. And this is important for practical things. And this is the other aspect of my life. So the thing over here is the Cardiff University logo, which I'm affiliated with. This is my startup, 5 Alarm Bio. And we're trying to apply this sort of logic to get therapeutics, because life does go wrong. Your biochemistry is going wrong right now. Ray's probably more than some of you. And this is an illustration of this. And this is the lens of the eye. This is on the left is from young mice. Can't take lenses out of young children. And on the right is a 67-year-old human. And that is his actual color. And any of you who've had cataract surgery will be able to actually see this yourself in the change in your color vision. The brown color is chemical crud accumulated in the lens. And it's not just accumulating in the lens, it's accumulating in all your cells. And that crud damages the cells, including damaging the crud removal mechanisms in the cells. And our hypothesis then is this leads to an exponential runaway and the cells stop working and you die. Death is considered an adverse medical outcome. And when I thought about this, I realized there was something you could do about it, and there's actually some molecules we can develop that will modulate this process. And so that's what we've done in the startup. And this is just one example of the sort of results we're getting. This starts out with, what is life, me eating chips, and ends up with developing drugs, which I think is quite cool. This is an experiment with this little guy at the top here. This is the nematode worm C. elegans. You're going to hear more about that later. And we're just tracking movement. So it's an automated vision system. We've got a dish of these little worms, and they're swinging around, and you track how many of them are moving. And that's what's plotted up the y-axis. And along here is the age of the worms. And the black line is the control worms, and they start out fairly young and healthy. There's the sort of teenage dip where they just can't be bothered to move. And then in adulthood, they peak, they're all moving, and then it drops off exponentially as they age. And the red line is what happens if you exactly the same worms, exactly the same conditions, treated with our prototype drug. This makes, in human terms, a 60-year-old worm rather as fit as a 45-year-old worm. And this is absolutely awesome. I'm incredibly excited that we could use this to develop treatments for diseases and disabilities of old age. Or I would do if we could get anybody to fund us, because investors don't seem to be interested in this. They want something about in AI. So if any of you know rich people who don't want to get old, please come and talk to me afterwards. That's it. Thanks very much to Sarah Seeger in particular on the left, who's been my inspiration and guru and guide in astrobiology. And Janusz, who's going to talk next, has been an awesome collaborator. 5 Alarm Bio team. To all the people who've said I'm talking nonsense, and please prove otherwise, and to you for your attention.